Dear students, in this session we will be discussing about complete dominance, incomplete dominance, codominance, multiple alleles, lethal allele, pleiotropy, epistasis and polygenic inheritance. So, first of all, we will discuss the meaning of certain terms which are generally used in these types of inheritance patterns. We know genotype. It is the genetic makeup of an organism, and phenotype is the physical characteristics of the organism which is encoded by the genotype. And for every gene, there will be different alleles. In a diploid organism, there will be two alleles for each gene, and the alleles may be either dominant allele or recessive allele. A dominant allele is a particular allele which is phenotypically expressed over the other allele, while the recessive allele is the allele that is only expressed in the absence of a dominant allele. It will be expressed only in a homozygous recessive condition. An autosomal trait is a trait which is located on an autosome, that is a non-sex chromosome. The homozygous condition is a condition where an individual will be having two identical alleles for a particular gene. The condition heterozygous means there will be two different alleles for a particular gene. So we will begin with the three types of dominance which are observed in genetics. They are complete dominance, where only one of the two alleles is expressed in a heterozygotic individual. The second type is codominance, where both alleles are equally expressed in a heterozygotic individual. And in incomplete dominance, there will be varying levels of expressions in heterozygotic individuals, and each individual may display an intermediate phenotype. Coming to complete dominance, we know from the experiments by Gregor Mendel on pea plants, where he used pea plants having a number of easily observable traits which were determined by two alleles. And all the traits on which he performed his experiments, all the traits he studied, one allele was dominant for the trait and the other was recessive. As a result, in Mendel's experiments, offspring always looked like one of their two parents due to the complete dominance of one allele over the other. The phenotype of the offspring from parents with different phenotypes always resembled either one of the parents. For example, in a cross between a red flower and a white flower, he always obtained a red flower. A homozygous red flower crossed with a homozygous white flower, the result was a heterozygous flower whose color was red, determined by the dominant allele, dominant red allele. The result is shown in the Punnett square here, while a homozygous red flower, capital RR, when crossed with a homozygous white flower, small rr, resulted in a heterozygous flower, capital R, small case r, which will have the dominant characteristic, dominant red color. But this is not always the case, because some genes display incomplete dominance. That is, individuals with heterozygous alleles exhibit a phenotype that is intermediate between those with the homozygous alleles. So this is incomplete dominance. And with incomplete dominance, a cross between organisms with two different phenotypes produces offspring with a third phenotype, which will be a blending of the parental traits. It is like mixing of paints. Mixing red with white will make pink. Neither red nor white will dominate the other. For example, in this experiment, when a plant having red flower is crossed with a plant having white flower, the progeny plants will be having pink colored flowers. So, when a homozygous red flower plant is crossed with a homozygous white flower plant, the result is a heterozygous plant with capital R and small case r as shown in this image. And this heterozygous plant will not be red or white, instead it will be pink in color in the case of its flowers. This is known as incomplete dominance. Next is codominance. As we discussed now, complete dominance is a situation in which one allele is clearly dominant over the other. And in incomplete dominance, neither allele dominates over the other. Now we are going to discuss the third type of dominance, that is codominance, where both alleles are simultaneously expressed in a heterozygotic individual. Human blood groups Mn and Mn are example for codominance. These blood groups are distinguished by the presence of two specific proteins, the protein M and the protein N, on the surface of RBC, red blood cells. Group M individuals will have one protein, group N individuals have the other protein, and group Mn individuals have both proteins on the surface of their RBC. A person's blood type, Mn blood type, is determined by alleles of a certain gene. The Lm allele specifies production of the M marker displayed on the surface of RBC, while Ln allele specifies the production of a slightly different N marker on the surface of RBC. And a homozygotic individual, LM, will have only M protein, and a homozygotic individual, LN, will have only the N marker on the surface of their RBC, while the heterozygotic individual, LN, will have both type of markers, both M marker and N marker, in equal number on their cell surface. So this is an example for codominance, where both alleles are simultaneously expressed in a heterozygotic individual. So this image shows complete dominance, where the color blue is dominant over the color yellow. So in a homozygotic BB condition, blue color is expressed. In a homozygotic YY condition, the Y color is displayed. And in a heterozygotic BY condition, the Y is masked and the blue color is dominant. In the case of codominance, homozygotic blue displays a blue color, homozygotic Y displayed a yellow color, and a heterozygotic blue and yellow individual displays a mix of both the blue and the yellow colors. In incomplete dominance, the homozygotic blue here is also blue, homozygotic yellow, Y here is also yellow, and a heterozygotic BY individual will display a color different from that of the individual colors. So it was expressed as green in color here. Now we will discuss multiple alleles. Although human beings and all other diploid organisms have only two alleles for a given gene on an individual level, there will be multiple alleles in a population level. As a result, different individuals in the population may have different pairs of these alleles. This could be easily explained by explaining the coat color gene in rabbits. The CC gene specifies coat color in rabbits, and this particular gene comes in 4 different alleles in a population of rabbit, even though each single rabbit can have only 2 alleles. These 4 alleles are capital case CC, CCH, CH, and small case CC. A diploid rabbit having the capital case CC allele will have a black or brown fur. A CCH rabbit have a chinchilla coloration. A CH rabbit has a Himalayan patterning, and a small case CC rabbit will be albino, having a white fur. So the wild type, first one is the wild type, will be having black or brown fur. The CCH chinchilla will be having a black tipped coat with white fur. And the Himalayan patterning is a rabbit having white fur with black paws, nose, ears and tail. And the small case CC rabbit is albino having a white fur. And this type of multiple alleles allows many possible dominance relationships. In this case, the capital case CC allele is completely dominant to all the other alleles. The chinchilla allele, CH allele, is incompletely dominant to the Himalayan CH and albino small case CC alleles. And the Himalayan CH allele is completely dominant to the albino small case CC allele. So this is an example for multiple allele, and there will be different types of dominant pattern among the alleles. As a result, different combinations of phenotypes could be observed in a population. Another example for multiple allele is the human ABO blood group. We know there are 4 possible phenotypic blood types for this particular gene: A, B, AB and O. According to the blood type, individuals can have the A carbohydrate for blood type A, the B carbohydrate for the blood type B, and both A and B carbohydrates for blood type AB, and there will be no carbohydrate, neither the carbohydrates will be present in a blood group O. So this blood group ABO are formed by various combinations of 3 different alleles: IA, which codes for carbohydrate A, IB, which code for carbohydrate B, and small case I, which code for the lack of any carbohydrate. So in a diploid individual, each individual have 2 alleles for this particular gene. An individual with 1 or 2 IA alleles will have blood group A. Those with 1 or 2 IB alleles will have blood group B. And those with both the IA and IB alleles will have blood type AB. And those with a genotype II, small case II, will have a blood type blood group O. Now we are moving on to lethal allele. Lethal alleles result in the death of an individual. Some genes have alleles that prevent survival when either it is in a homozygous condition or in a heterozygous condition. As a result, there are different types of lethal alleles: recessive lethal, dominant lethal, and conditional lethal. An example for a lethal allele is the lethal yellow allele, which is due to a spontaneous mutation that makes the coat color yellow in mice. Mice that are homozygous to this allele, AY genotype, die early in their development. So this is a situation where the allele becoming lethal in the case of a homozygotic condition. So there are different types of lethal alleles. They are recessive lethals, dominant lethal allele, and conditional lethal allele. Coming to recessive lethal allele, a pair of identical alleles that are present in an organism that ultimately result in the death of the organism are known as recessive lethal allele. These types of lethal alleles are fatal or result in the death of the animal only in the homozygous condition. Example is homozygous achondroplasia. Next is dominant lethal alleles. These are alleles that need to be present in a single copy to be fatal. And generally these alleles are not commonly found in populations since it results in the death of the organism before it can transmit its lethal allele to its offspring. The example is Huntington's disease, which is a rare neurodegenerative disorder. Next is conditional lethal allele. These are alleles that will be fatal in response to some environmental factors. Example is favism, which causes the carrier to develop hemolytic anemia when they eat fava beans. Now we will discuss pleiotropy. Sometimes one gene may have multiple effects, and this is known as pleiotropy. For example, albino individuals lack pigment in their skin and hair, and they also have crossed eyes at a higher frequency than pigmented individuals. Another example is sickle cell disease. The symptoms associated with sickle cell disease. Individuals with a sickle cell disease are homozygous for a mutant allele, which result in a sickle-shaped red blood cell in their body. Because the sickle cell-shaped red blood cells can deliver less oxygen to the tissues, sickle cell disease have many pleiotropic effects. Symptoms include pain in the bones, especially in the back, the long bones and chest. And as the disease progresses, additional symptoms develop. For example, fatigue, paleness, rapid heart rate, shortness of breath, and yellowing of the eyes and skin, or rather jaundice. This situation occurs only in individuals who are homozygous for this particular mutated allele. While people having heterozygous condition, having only one copy of this allele mutated for the sickle cell mutation, will not have the symptoms of sickle cell anemia. Another example is a human genetic disorder known as Marfan syndrome, which is also caused by a mutation in one gene. Yet, the single mutated gene affects many aspects of growth and development, including height, vision, heart function, etc. As a result, the individuals will have unusually tall height, thin fingers and toes, dislocation of the lens of the eye, and also heart problems. So, pleiotropy is a condition where one gene affects multiple characteristics. Next is epistasis. This is a gene affecting the expression of another gene, controlling the expression of another gene. Sometimes a gene at one location on a chromosome can affect the expression of a gene at a second location. This is epistasis. Epistasis takes place when the action of one gene is modified by one or several other genes. These effector genes are known as modifier genes, and the gene whose phenotype is expressed by this modifier gene is known as epistatic, while the phenotype which is altered or suppressed is known as hypostatic. The epistasis is seen at both genomic levels and phenotypic levels. At genomic level, it is observed where under certain conditions, one gene codes for a protein that prevents the transcription of the other gene, regulating even the rate of transcription of another gene. And at the phenotypic level, examples include the gene causing albinism, and this gene will hide the gene controlling the color of a person's hair. A classic example for epistasis is the genetic interactions that produce coat color in horses and other mammals. In horses, the brown coat color, capital case B, is dominant over the tan color, small case b. And there is a second gene which can control the deposition of pigments coded for by these genes in the hair. So the gene expression by these coat color genes is dependent on a second gene which controls the deposition of pigment in hair. So the dominant gene, capital case C, codes for the presence of pigment in hair, whereas the other gene, the small case c, is recessive gene and it will code for the absence of pigment. If a horse is homozygous recessive for the second gene, small case CC, this horse will have a white coat regardless of the genetically programmed coat color, whether even if it is capital case BB, the produced pigment will not be deposited in the hair, and as a result, the horse will be having a white coat color. So this is epistasis, a gene affecting the expression pattern of another gene. Now, we will discuss about polygenic inheritance, or rather the effect of multiple genes. Polygenic inheritance occurs when one characteristic is controlled by two or more genes. Several features in human beings like height, eye color, hair color, etc. comes in lots of slightly different forms because they are controlled by many genes. Each of these genes will be contributing to some amount to the overall phenotype. Likewise, in wheat kernels, there are three genes that make the reddish pigment in wheat kernels. They are A, B, and C, and each gene comes in two alleles. The capital case A, B, C makes pigment, and the smaller case A, B, C does not make the pigment if they are the alleles. The peculiar factor is that these alleles have additive effect. The small case AA genotype would contribute no pigment. The capital case A, small case a genotype would contribute some amount of pigment, and the capital case AA genotype would be contributing more pigment. The same pattern is for the B and C genes. A similar level of pattern is observed in human skin color also, as can be observed from the figure given here. There are three genes A, B, C, which will come in a capital and a small case forms, and according to their allelic pattern, they will be contributing more or less amount of pigmentation on skin, resulting in various skin color patterns in a human population. So, what is the difference between pleiotropy and polygenic inheritance? In both cases, there are more than one gene influencing a same phenotype, same characteristic. But in the case of pleiotropy, one gene affects multiple characteristics. One gene controls the expression of other genes. Either it will activate the expression or it will suppress the expression of the other gene. While in the case of polygenic inheritance, one trait is controlled by multiple genes. Each gene is having some contribution towards the final phenotype. Marfan syndrome and sickle cell anemia are examples for pleiotropy, while skin pigmentation is an example for polygenic inheritance. So, we were discussing about different types of inheritance patterns: complete dominance, incomplete dominance, codominance, multiple alleles, lethal allele, pleiotropy, epistasis, and polygenic inheritance. Thank you so much for listening.